So to kind of get you resituated as to where we are in biology. The ones I have checked here were our first unit, so talking about really large things such as the Earth and organisms as a whole. And then we've already discussed atoms and molecules, which is part of this unit and will be on your test. So the next tidbit that we're going to run into are organelles and cells. Now, we already talked about these in lab, so I am going to very, very briefly go through these again. But again, we went through it more in detail in lab, so I'm not going to spend much time on it here. These are still posted in the PowerPoint, and you can open up the PowerPoint separately to review them. So recall that organelles are just a bunch of organic molecules. Those organic molecules, our lipids, our fats, our carbohydrates, come together to form organelles. So each organelle has a very specific function. We've talked about the nucleus and the cell membrane. Here we have chloroplasts in our plant cells, and we know that a chloroplast has very different functions than, say, the nucleus. So here, recall that we have our plant cell. A major giveaway is that it's green. But if you didn't have a color picture, you should be able to know that, okay, we have very straight sides here. That support is from this cell wall. So therefore, it must be a plant because animals don't have a cell wall. So both plant and animal cells have a nucleus. This is where the DNA is stored, and this is what controls the cell. Both plants and animals have mitochondria. This is where ATP is created. So recall, this is where cellular respiration is happening, because cellular respiration is what gets us ATP. You'll also hear the mitochondria called the powerhouse, because it's like a power station. That's where all of our electricity comes from, is from the powerhouse. Both plants and animals have ribosomes, even though the plant isn't labeled here. The way to identify this structure are just these really small dots that you see. Just to recall, the nucleus is going to be very central, very large circular object, and mitochondria are kidney bean shaped with lots of squigglies or lines in them. So ribosomes are where proteins are being made. So ribosomes are made of RNA. So one and the same. We learned RNA makes proteins. Well, ribosome is a type of RNA. And then only found in plant cells are chloroplasts. Recall that this is where photosynthesis is occurring. Both plant cells and animal cells have an endoplasmic reticulum, or you'll see this abbreviated ER for short. And this is what's going to move proteins around the cell. The proteins are typically made in the cytoplasm, so the empty area between organelles, but they're usually not needed there. They're needed elsewhere. So the endoplasmic reticulum gathers them and delivers them to where they need to go. And both plants and animals have this. Now in lab, when you were doing your activities, you learned about the smooth endoplasmic reticulum and the rough endoplasmic reticulum. Everything that is in these two slides are what you're responsible for on the test. On the lab quiz, you're responsible for what's listed in the lab book. So there's some overlap, but for the test, since we have so much on the test, I decided to keep it a lot simpler. Both plant and animal cells have Golgi bodies, so these are going to package proteins to leave the cell. So endoplasmic reticulum transports them in the cell. Golgi body gets them ready to leave the cell. Both plants and animals have a cell membrane. So we've talked about this a lot, and we'll talk about it more here in the next few slides. But this is what's going to control what leaves the cell and what enters the cell. And finally, this is only found in plants, is the cell wall. And that's what's giving all of this support and structure to a plant cell. That's why a plant cell has such straight sides, because the cell wall has a lot more support than the cell membrane. So we've learned about all of these different molecules that have a lot of different functions. Now, cells benefit from most of those functions. So if a cell needs protein, it gets protein, but it doesn't grab it. There's this problem where cells need these molecules, but they can't easily just say, oh, I need protein, and it goes out and actually grabs protein. So what we're going to talk about next is how molecules get into these cells and also leave these cells. Because don't forget, cells make things. So our cell membrane, again, both plants and animals have these, and this surrounds all of our cells. It's called semi-permeable, so this term here. So the semi means partly, and permeable means to let things in. So it lets some things in, but it doesn't let everything in. Think about a screen door. A screen door is still going to let air in and water in and really small things in. But the little squares on a screen are pretty small. It's not going to let really large bugs in or anything like that. Our cell membrane is the same thing. It lets some things in, but not everything. So something to keep in mind, and we're going to see this theme a lot in the next few slides and in this lecture, is that cell membranes are in a solution. It's in something that is liquid. Yeah, our skin cells are exposed to the air, but within our body, it's constantly surrounded by liquid, whether it's blood or interstitial fluid. That kind of fluid is just, it's not blood, it's just some sort of solution. It's a liquid that lines all of our cells. And this is really important to keep in mind when we talk about how different molecules enter and leave the cell. So what is a solution? So a solution is anything that's dissolved in a liquid. Now, that thing that's dissolved in a liquid, it can be all sorts of things. It could be things like salt or sugar or oxygen. It doesn't have to be a solid. So we think of solids such as salt or glucose. That's a solid dissolved in something. But it could also be oxygen or carbon dioxide, so other gases. They could also be liquids that are dissolved in another liquid. So again, a solution would be this whole beaker. So multiple things mixed together. Whereas a solute are these little yellow balls in here. This is what is being dissolved. So if you think about salt water, the salt is what's being dissolved. Whereas the solvent is what things are being dissolved into, and typically it's water. All of the examples that we're going to use in this class is water. Most of the examples in life itself is water, so that's why we're going to stick to that. Know that other things can be solvents, but we're going to use water for this course. So again, this whole beaker would be the solution. It's the total. The solute is whatever particles being dissolved, whether it be a gas, solid, or liquid. And then the solvent is always going to be a liquid. And in this class, we're going to talk a lot about water. So if we have a solution, so say a solution is 100%. So whatever the solute is, plus whatever the solvent is going to be 100%. So let's kind of illustrate what I mean here. So let's say I gave you a word problem and we were talking about a 30% sucrose solution. Now remember, solution means the solute plus the solvent. It's the entire solution, the entire liquid. Now if I say 30% sucrose, that's referring to the solute. I'm saying that in this solution, I have 30% sucrose. So here's the solution down here. We're saying of this entire thing, 30% of it is sucrose. So what would that make the liquid? Hopefully you were thinking along the lines of around 70%, because we know that the solute plus the solvent is going to give us 100%. So this liquid is going to be 70%. So that's saying in this solution, 30% of it is sucrose, and 70% of it is water. So now that we understand solutions, and particularly solutes, remember that's what's being dissolved, we can talk about how cells get these solutes, because that's what cells are looking for. Cells need those solutes like oxygen, or glucose, or salt, or other ions. And all of this is suspended in a liquid, whether it's water, or the interstitial fluid, or our blood. So everything in our body is a solution, and our cells need those solutes. So one way that our cells can get these solutes is through passive transport. Now what passive transport means is that we don't need to use energy. And by that I mean we don't need to use ATP. The cell does not have to go through any effort in order to get that molecule inside of the cell. So here again, here is our solution on the outside of our cell. So here we have the liquid, and then we have these solutes that are floating around that our cell needs. So one way that these cells can enter is pretty easily. They don't need energy to do it. Now there's two different types of passive transport. There's diffusion and osmosis. So one form of passive transport is diffusion. And you may have heard this term because it applies to a lot of things outside of biology. So in diffusion, molecules are moving from a high concentration to a low concentration. So take a look at this beginning square. Here on the left-hand side, we have a whole bunch of solute. Let's say this is glucose. So we have a very high concentration on the left-hand side. Well, here on the right-hand side, we don't have any, meaning that's a very low concentration. Well, naturally, any kind of molecule, whether it's a solid, such as glucose, a liquid, such as water, or a gas, such as oxygen, is going to move from high concentration to low concentration. And it doesn't need energy to do that. It's fairly easy for it to do. Now, this difference in concentration is considered the concentration gradient. So if we're going from high to low, that's saying we are going with the concentration gradient. We're going with the flow. We're not doing anything difficult. We're going from something very high to something very low. And over time, it equilibrates. So we see here in the very last picture, we have about the same amount of glucose on one side of this membrane than we do on the other side. So you've seen diffusion before. You've seen it or experienced it a lot of times in your life. So here I have a candle. So those scent molecules and those scent particles are really, really concentrated right next to the candle, whereas not so much on the other side of the room. But when you light that candle, those particles start spreading from right at the candle, and they spread out. They're spreading to areas of low concentration. That's why you can light a candle or spray something in the room or anything like that, and you'll smell it in other areas because those particles are diffusing to areas of low concentration. If you take a glass of water and don't stir it at all and just drop in say food coloring, over time it will completely fill this glass without stirring, because that food coloring is going to move from it's going to move from high concentrations to low concentrations. An example in our body is the lungs. So here's a little lung pocket. Here is a blood vessel. Our lungs have tons of oxygen in it. Our blood doesn't. So oxygen is going to move from this high concentration in the lung to a low concentration in our blood. Our blood is carrying carbon dioxide, so it's a really high concentration in the blood and low in the lung. So when we inhale, we're taking in oxygen, and when we exhale, we are releasing carbon dioxide. So we're going to go ahead and stop there before continuing on to part 3.